Cadmium is actually a type of metal, so it's very useful in modern industry. For example, in battery industry, in electronic device, and also in painting. So it's very important in industry. But however, cadmium is also toxic metal, so it has been associated with the occurrence of a variety of diseases. For example, cardiovascular disease, cancer, diabetes. So it's widely distributed in our environment. For example, in soil, in the air, and also in our food, for example, mushroom, shellfish. Very critically and importantly, so the effect hasn't been well studied in animal models, in human subjects, and in other words, so it hasn't been well demonstrated in the in vivo level. So we found that cadmium exposure can increase the activity for some drug transporters, and then we, based on those preliminary studies, we did further studies and found that cadmium exposure could have a broad impact on drug disposition and response. By gathering such knowledge, so eventually, so we will be able to understand that, a wide variety of clinical drugs, so their clinic treatment and therapy and their the response among patients would affect by cadmium exposure. When we began the study, so we think cadmium is actually a substrate for our transporters. So then we think that cadmium may be able to competitively reduce the activity for the transporters we're studying. So the student, he found that cadmium exposure can actually increase the activity of these transporters. So the lesson we learned is, and also I think it's very important for students to have this kind of lesson. So sometimes when you obtain a result that is opposite to what you're expecting, it's not necessarily bad, because you can start working from there and actually it can turn out to be positive.